So you found the video. This is the lecture replacement. At this point in class, you should have already received some worksheets from the substitute. If not, please go ask the substitute for your worksheets now. You may want to start those worksheets as you watch the video and as you take your notes. So again, if you have not gotten those yet, stop the video, go talk to the sub, make sure you get the worksheets that you need. It's expected that you'll get some good notes from this video. You will finish those worksheets, give them your best shot. We'll go over them in class next time, and so they need to be done by the next time I see you. So next time I see you in class. All right. So today's content is on Mendelian genetics, and then some contradictions to what this guy Gregor Mendel came up with. So first of all, what is Mendelian genetics? So this is our guy Gregor Mendel, and he's credited with basically coming up with the foundational concepts of genetics that we still use today. Next week, we'll talk about why he was a filthy liar and made up some of his data, but that's not for today. Today, we'll talk about the things that Mendel got right. So first of all, Mendelian genetics is the assumption that individuals inherit two copies of each trait. We now call those alleles. He was the first person to really concretely say that the alleles are different from each other. In other words, if they're heterozygous, only the dominant allele is seen in the phenotype. The recessive allele is still there, but is masked by the dominant allele, so you don't see it, even though it's still there. And those core ideas provided the foundation of modern genetics that we still rely on today, even though there are some pretty serious flaws. So, really quickly, I'm going to just give you a really, really short story of how Mendel came up with his ideas. We're not going to go nuts with history here. What you need to know is that Gregor Mendel was basically, he grew a ton of peas at his monastery. And he noticed that the pea plants could produce different colored flowers, among other traits. And so what he did was he started breeding different pea plants with each other. So he took a plant that made purple flowers and a plant that made white flowers. And then he would take some pollen from one, transfer it to the other with like a paintbrush or something, very carefully, and then see what kinds of plants were produced in the next generation. And so when we talk about this kind of thing, we're, again, like we said last week, we're discussing what's called a monohybrid cross, looking at one trait and hybridizing them. And so you need to get used to this terminology. So the plants that you start with, we call those the parents or the P generation. Their offspring would be the F1 generation. And then if you were to take two members from that generation and cross them with each other, that would be the F2. So here's what Mendel did. He took some purple flowering plants and some white flowering plants. And when he crossed them with each other, all of the offspring were purple flowering plants. So take a second, consider what we now know that to mean. We now understand, thanks to Mendel, that purple allele would have been dominant. And so we would have only seen purple flowering plants. And then what he did was he took those purple flowering plants and he crossed them with each other. And in the next generation, this is the F2 generation now, sorry about this copy, I don't know what happened here. About 3/4 of the plants had purple flowers, and about 1/4 of the plants had white flowers. And so Mendel needed to explain how that white flowering trait could disappear for one generation and then come back in the next generation. So he had some ideas. His first idea was that each trait will have different versions. We now call those traits genes, and we now call those versions alleles. So that's what we call them today. He didn't have that terminology then. So for each gene, there are different versions that we call alleles. For example, the white and purple allele. And then, for each characteristic, an organism will inherit not one copy, but two copies, one from each parent. And we now use this terminology and we say that if the alleles are the same, that's homozygous. If the alleles are different, that is heterozygous. And just another reminder, that is the genotype. So these are two of Mendel's big ideas. And then his third idea is that, and we said this before, if the alleles are different, you only see the dominant allele. And the recessive allele is present, but it is masked by the dominant. So there's no noticeable effect. And then that leads us to the phenotype. So the physical appearance, the phenotype will only show the dominant allele, whereas the genotype gives you the combination of both alleles, whether or not they're dominant. And then one of Mendel's other big ideas is this idea of the law of segregation, which is that during the production of gametes or during reproduction, each parent only gives one copy or one allele to their offspring. And these ideas today don't sound like a huge discovery, but at the time these are big ideas, and so we still discuss Gregor Mendel as an important figure in genetics. So go back and rewatch any of that if you need to get your notes down, and I'm going to carry on away from Mendel's big ideas. The last thing that you'll need to know is that Mendel studied multiple traits, right? You don't need to memorize these traits. We don't really care too much about that. But you do need to understand that since Mendel was studying more than one trait, he could also look at dihybrid crosses. Remember, dihybrid crosses from last time would be two traits at the same time. So we now understand that for homologous chromosomes, we're going to have multiple genes present, and each one will have different alleles. And so what Mendel did was he was understanding that if there are two traits at once, or a dihybrid cross, his idea was that those two traits would not influence each other. So, for example, peas can be yellow or green. Let me clip back here. Peas can be yellow or green. They can also be wrinkly or they can be round. So, his idea is that just because a pea happens to be yellow, it doesn't also have to be round. It could be yellow and wrinkly, or it could be green and round or green and wrinkly. These two traits don't actually influence each other, and that is his law of independent assortment. So that sets that each pair of alleles segregates independently of the other pairs during gamete formation, and so that the presence of any allele should not affect the inheritance of a different trait, right? And that's the law of independent assortment, and you are actually putting that into practice on your worksheet without knowing it with dihybrid crosses. And then again, next week, when we study the HL content, we'll learn that this is actually a big lie and that Mendel just threw out the data that he didn't like. But that's for next week. Okay, that's the law of independent assortment. Okay, here is our new information. So the new stuff for today starts now. Everything else we talked about is what we did last week. So the other thing that Mendel did, and this is quite easy, is he did what's called a test cross. And what you do for a test cross is, let's say you have an individual who's showing the recessive phenotype, and you know they're homozygous because if they're showing the recessive phenotype, they have to have two copies. So what he did was he was taking homozygous recessive individuals, crossing them with something that was showing the dominant phenotype. But if it's the dominant phenotype, you don't know if they're homozygous or heterozygous. And so he would cross them to test to find out. So here's an example to sort of demonstrate how the test cross works. Let's say we have a purple flowering plant and we're crossing it with a white flowering plant. Remember that the purple flowering plants could be homozygous dominant or they could be heterozygous. The white flowering plants, this is the recessive trait. And so we know that the white flowering plant has to have two recessive alleles. So here's our white flowering plant. That purple flowering plant has at least one dominant allele, but we don't know what the other allele is. We just know that it's purple. So then what you do is you'd run a test cross and you'd count the offspring. And if about half of the offspring produce purple flowers and the other half are white, well, then you know that down here, this has got to be our recessive allele. Because when you fill this in, then you're going to get recessive homozygous. And this gives us our heterozygous, and then 50% are going to be dominant and 50% are going to be showing the recessive phenotype, right? So half purple, half white. So taking these two plants, and we don't know their genotypes, crossing them, counting up the offspring, that is the test cross. So that's how that works. We'll revisit this next week too. That's pretty simple, I think. That's a test cross. So the question would say, what are the genotypes of the parents and the offspring? So, consider doing a test cross with humans. Obviously, that would present some ethical concerns. Yeah, so we don't do test crosses with humans. Rather, we do what's called a pedigree. So, with a pedigree, you have to basically know your family history. And then you can track the inheritance of traits through multiple generations. And so a pedigree is set up like this, where you look at two parents and their offspring. The squares are supposed to be male, the circles are supposed to be female. And then the shaded shapes, that doesn't mean that they have dominant or recessive alleles necessarily. All it means is that they're showing the trait that you are interested in. So this male is showing the trait. Now, that trait could be blue eyes or brown hair or God knows what. It could be sickle cell anemia. But this is just showing the trait, and this person does not have the trait. So I'd like to see if you can kind of work this out. Take a second. Look at the parents here, the offspring here, these parents, these offspring. The trait that we're studying in this pedigree, is it controlled by a dominant allele, or is this trait controlled by a recessive allele? Take a second, pause the video. Seriously, pause it. Pause the video and see if you can work out if this trait is caused by a dominant or a recessive allele. Pause it. Okay. So, let's take a second. If we see two parents that do not show the trait, and then they have a child who does show the trait, we know for certain that this has got to be recessive. And here's why. If we consider, let's say, for example, we had it wrong. We think this is dominant. This kid, number 4, must have picked up a copy of the allele from somewhere. So if this kid, number 4, has the allele and they got it from mom or dad, and if we assume it's dominant, that means mom and dad must have that allele and they must be showing it. So that's not possible. So we know this kid, I'm just going to use the letter A here. This kid is homozygous recessive. And that also means that the parents must have one copy of the recessive allele each. So mom's got to have a heterozygous genotype and dad must have a heterozygous genotype. And if you didn't follow that, basically, number 4, that child has to get one recessive allele from each parent. And they got one from dad, and they got one from mom. And they can only do that if this trait is recessive. Okay, so that's how you can use a pedigree to work out if a trait is dominant or recessive. The substitute gave you a pedigree worksheet. I'll leave it up to you how you want to do this. I'd recommend pausing the video, knocking out a few pedigree problems, and then coming back to the video and finishing up your notes just to check if you get this concept. Okay, so if we assume that we're moving on now with notes, IB wants you to know 2 examples of traits that are controlled by either a recessive or a dominant allele. So you need to memorize that cystic fibrosis is a disease that affects the airways in your lungs. Basically, you build up this sort of extra fatty tissue in your lungs, and it makes it hard to breathe. And so cystic fibrosis is caused by a recessive allele. IB wants you to memorize that. IB also wants you to memorize that there's another condition called Huntington's disease, and this is an example of a disease controlled by a dominant allele. And so this one is a bit more dangerous because if you inherit just one copy of the allele, then you are going to get Huntington's disease. It's also a deadly disease that shows up late in life after you've already had kids, usually. So it's quite sad because you've already had kids and then you develop Huntington's, and then you're thinking whether or not you've given that dominance allele to your kids, and it can be actually quite tragic. But that's Huntington's disease. IB wants you to memorize that it is caused by a dominant allele, whereas cystic fibrosis is caused by a recessive allele. Just basic memorization on that one. Okay, and that brings us to our last concept. So everything we've talked about so far has been Mendelian genetics. There's no blending of traits. You're either dominant or recessive, and there's no in between. But in reality, we have to understand that genetics is way more complicated than that. So our one example that we'll do today, and this is the last thing we'll do in this lesson, is this idea of co-dominance. And this is where neither allele is dominant and neither is recessive, but rather, if you're heterozygous, you will show both alleles at the same time. Blood typing is a great example of this.